Hi, my name is Assaf. I'm a Master of Science degree student from Ben-Gurion University in Israel, and I'm happy to share with you my research about cell communication. Many mechanisms exist for cell-cell communication. Here we decided to focus on long-range mechanical cell-cell communication through the extracellular matrix, the ECM, as a model system to systematically study the different stages of cell-cell communication. Presented here is an example of a 3D projection live imaging video of a mechanically communicating pair of fibroblast cells, forming a band of dense fibers. Understanding and quantifying this communication could be very useful for applications where the cellular microenvironment and the interactions between cells and the ECM are established hallmarks, such in cancer or fibrosis screening. Current image-based phenotypic drug screening is performed with a trade-off. On the one hand, 2D or 3D cell imaging enables high-content, quantitative analysis of cell morphology but suffers from low physiological relevance. On the other hand, 3D spheroid screening is more physiological relevant but limits the measurements to extreme functional readout, such as cell viability and proliferation. Here we try to balance between the two. We use 2D finite element simulations of contracting cells surrounded with a network of connected fibers, and 3D experiments of live confocal microscopy imaging of gel-embedded fibroblast cells with a dual-channel imaging setting of GFP actin and fibrous channels. Both simulations and experiments included single cells and cell pairs. We defined three hallmark stages of cell-cell communication. One cell releases a signal. The second cell receives the signal, processes it, and changes its internal state in response. These cells feel, respond, and remember the mechanical properties of their microenvironment, and apply traction forces on their surrounding matrix, leading to ECM deformations and to a visible band of dense fibers. An ECM region located along the connecting axis experiences forces exerted by both cells. We divided these forces into two: the force exerted by the nearby cell and termed cell-ECM interaction, and the force exerted by the neighbor cell, termed the cell-ECM cell communication. The latter is an absent force in single cells. Before we quantify, to easily measure fiber density in between cell pairs in 3D, we transformed the microscopy axis to a new 3D coordinate system. Cells were segmented using the GFP actin channel and tracked manually. Fiber density in experiments was measured in 3D, based on pixels intensity of the fibrous channel. Fiber density in simulation was measured in 2D, by calculating the inner length of the fibers. Quantification windows were placed in the inner regions of a cell pair, or all around single cells. Now it's time to quantify. Cell-ECM interaction was measured in single cells and was identical between simulations and experiments. When the second cell joined, the case was different. Based on the difference between single cells and cell pairs densification, we could quantitatively decouple the contribution of the cell-autonomous and cell-cell communication components that together form the visible band of dense fibers between the cells. Of course, this is a dynamic process. In simulations, we configure the cells to contract independently for up to 50 frames. Experiments were imaged in two temporal resolution settings of 15 or 5 minutes between consecutive frames. Quantification windows were placed and measured adjacent to cell boundaries at all time. Seen here are the fiber density dynamics, time series, of the two communicating cells on the left. Then, we derivate our measurements temporally and correlated them using Pearson correlation. For this example, the correlation was 0.71. Our method was used for a variety of analysis, from basic communicating partners correlation through distinguishing between different pairs of communicating cells, testing it on pairs with no visible band, validating our results against manually created fake pair of cells, and even recognizing asymmetric interactions of leader and follower roles, using cross-correlation in pre-configured simulations and with advanced tools such as Granger causality and experiments. What was mainly surprising was the discovery of decoupling between fiber densification and the ability to recognize communication, which was possible only after shifting the quantification window slightly above or below the axis connecting the communicating cells. To ultimately test the sensitivity of our method, we performed an analysis we termed as matchmaking. Here is a short video about it. We first found all cell pairs in each experiment, as mentioned earlier. For each cell pair, we placed two quantification windows in the inner regions between the communicating cells, adjacent to cell boundaries and following their movements. We quantify the fiber density dynamics of each cell and temporally derivative the time series. Then, we separated the partners. For each cell, we considered all other cells from all other cell pairs in the experiment. For each of the optional partners, we correlated the fiber density dynamics and saved the result. Finally, we ranked the correct partner correlation in relation to all other correlations. So, in case it was the highest, its rank was 1, which means we successfully matched the partners. Even though the probability of matching between communicating partners was as low as 0.02, we were completely correct, highlighting the high sensitivity of our approach, even for pairs that did not form a visible band of dense fibers. Our method sets the stage to measure the fundamental aspects of intercellular long-range mechanical communication in a physiological context, suggesting a new signature for mechanical cell ECM cell communication. This research was performed under the supervision of Dr. Assaf and is a joint project with Ayelet Lessman Lab at Tel Aviv University. The research was submitted to bioRxiv Library. These days, I'm looking for my next career challenge, and will be happy to hear about available opportunities. Thank you for listening.